So the purpose of today's video is to find out the correct meaning of the Genbank entry and how to make the sense out of Genbank entry. So in we will go to the URL www.ncbi.nih.gov/Genbank. Yes, and when we move, it will take us to the home page of Genbank, which is a which is a database which contains of annotated collection of all publicly DNA sequences. It is a database of National Institute of Health. So here we can see that in the search box left side, it is pull down menu we have selected. We can select from here nucleotide. We want to search for nucleotide sequence. And in the right search box, we can type here the name of any nucleotide sequence with GenBank ID. For example, we use here X01714. So when we search, so in the results page, it will display a short definition for the nucleotide sequence entry. So here there are many formats. We can see this as the result page, and we can see that here there are many formats for display like summary, then summary, Genbank full, FASTA text, graphics, ASN.1 division, history, accession list, GI list, etc. Now we have, when we click on summary, it will display a short definition for the nucleotide sequence entry we have entered for the ID X01714. Now we want to see, we want to make sense of Genbank format, Genbank ID. So what we will do that we will select Genbank, then it will again take us to the Genbank page. We will see that there are many fields which have been displayed here. We will understand the meaning of each and every field one by one. So, first of all comes the locus. What does this locus means? It is written here X01714 BP means base pairs DNA linear BCT this much. So this gives us the locus name. Locus name is X01714, which is an arbitrary name, and the size of this nucleotide sequence is 1609 base pairs DNA, means this DNA sequence or nucleotide sequence is this much length. And again it gives us the nature of the molecule, which is linear. And again now we go to the definition section, and this is the name of the molecule which on when it has been deposited. This is the date. Now in the definition it is given that E. coli DUT gene for DUTPase. So this gene entry, which nucleotide entry which we have entered, is the gene entry for DUTPase enzyme, and it is present in E. coli, and it provides this definition provides a short uh, definition of the gene that corresponds to the entry sequence. So here it is the E. coli DU TPase gene. Now the third line is accession. Accession is the lists the accession number, and this accession number here is X01714. It is a unique identifier within across various databases such as the protein sequence databases, and thus we can see that X01714 is the accession number. Now the version, we come to the version file fills you in on the synonymous of past ID numbers. So the past ID numbers which is synonymous to them are, and the version is this X01714.1. This, it is the first version. Then comes key organism is E. coli. Yes. Then comes the keywords DUTPS uni unidentified reading frame. So this keywords introduces a list of terms that broadly characterizes the entry. So you can use these terms as keywords for certain database searches. For more on using keywords in database searches, you can use PubMed as well. Now then comes the source. Source here is E. coli. So the source divulges the common name of the relevant organism to which the sequence belongs. So this is the name of the relevant organism, and to this the sequence entry we entered belongs. Then comes the organism. Organism is E. coli, as we can see, and it gives a more complete identification of the organism, complete with its technical taxonomic classification. So bacteria, proteobacteria, gamma proteobacteria, enterobacterial. So this is the whole taxonomic classification, that how E. coli has been placed. So then we come to reference section. So reference here introduces a section where the credits of the sequence determination are given. For example, here it has been mentioned 1 basis 1 to 1609. So different parts of the sequences can be credited to different authors. The difference section contains multiple parts such as authors, title, journal, and PubMed. So this is all the part of the literature section from which this entry has been taken from. Then the comment section comes. It introduces the last line of the top of the X01714 Genbank entry, and it contains pre-formatted text such as acknowledgements or information that doesn't fit in the previous sections. So then comes the middle section, which is source. Here, source indicates the origin of the specific regions of the sequence. Here, 1 to 1609, these are the specific regions of the sequence, and this is useful when we want to distinguish cloning vectors from the host sequences. So, in X01714, a 35 region is indicated. Here, it is indicated, and from position this from position 286 to 289. Yes, and in this promoter, these are the regulatory sequences. These are the promoter. It shows the precise coordinates of the promoter element, and it introduces the line that contains the coordinates of another promoter element. We can see here these are the promoter elements, and from the another promoter element is from 310 to 316. Here it is 286 to 291. Then comes miscellaneous feature 322 to 316. What does it mention? So it indicates the putative location of the transcript start messenger, that is messenger RNA synthesis. So for example, for X01714, this position is from 322 to 324. Now then comes ribosome binding site in regulatory. So it is the ribosome binding site which indicates the location of the last upstream element. Last upstream element was 324. So, so for here, the location of the last upstream element, that is ribosome binding site, is from 330 to 333. So significance of all the bases it is showing here. Now then comes CDS. It stands for coding segment, which introduces a complex section that describes the gene's open reading frame. Here we can see the gene's open reading frames are there. So there are other miscellaneous features which follow the CDS section. You can scroll down and see. Again, CDS section is there. Yes. So again, when we scroll a little bit lower, we can see the whole DNA sequence, nucleotide sequence, which we have of X01714, which we have entered. Similarly, we can take out the different formats we want to see from here. If we want to see FASTA format, which we used at number of times in order to run blast or take out this for the protein structure determination, then it comes over here. So from here we can scroll down the other ASN1 and different other file formats according to our need. So today we have been able to make out sense of the Genbank sequence, Genbank format, and this way we can understand that how the different fields in this very Genbank format, a particular nucleotide sequence is can be understood. Thank you.